Great, just share screen. Full screen. Right, great. So this is a recording of the first lecture in the antibody biotechnology module, the nature of immunity. So although the focus of the module is very much on antibodies and the use of antibodies in biopharmaceuticals, in therapies and things, it's important that we have an idea of the nature of immunity, because antibodies are a part of what we call the adaptive immune system. There's two types. There's the innate and the adaptive, but they're closely related. So it's important that we understand the basics behind the nature of immunity. Okay, in terms of recommended reading, right, this is not a course on immunology. It's a course, it's a module on antibodies. So we don't expect you to go out and buy books on immunology and things, but there are relevant parts of certain books which will be very useful for this module, and you can access through the library at the University of Nottingham the instant notes in immunology, this BIOS book by Lydiard and co-workers. There's also Reut's Essential Immunology, again, which you can access online through the library. And there's also other online resources as well, and various links. Right, the nature of immunity. First of all, we'll consider the causes of infection. Secondly, immune system components. And thirdly, the immune response. Now, alongside this recording of the lecture, we think it'd be very useful if you also considered this in conjunction with the video on immunity, which is produced by Elsevier. Now, on the Moodle, where it lists the lectures and recordings from the lectures from this module, we put a link in directly underneath the listing for this recording to the Elsevier recording. So do look at that. It's a very good description. So please look at that in conjunction with this recording here. Right, so first of all, the causes of infection and the infective organisms: viruses, bacteria, fungi, protozoa, and worms. And of course, there's various agents and toxins associated with these infective organisms. And the mechanisms of infection include invasion. Viruses and bacteria can infect cells, examples poliovirus, and of course COVID. excessive bacterial growth in body tissues. An example is pneumonia. And then toxins. Examples include the endotoxin from salmonella, which causes food poisoning, or the exotoxin, such as neurotoxin, secreted from Clostridium botulinum. So endotoxins and exotoxins. And healthy tissues are devoid of pathogenic bacteria and viruses because there are immune systems in operation. Okay, so that's the purpose of the immune system. Right. So this just lists some examples of infectious diseases. These are some of the main infectious diseases which come from bacteria, fungi, parasites, and viruses. And the ones highlighted in yellow are intracellular. Right, part 2, immune system components. So how is infection prevented? Well, first of all, by physical and chemical barriers, stopping the pathogens getting in the first place. This is where skin comes in, mucosal secretions of, for example, the alimentary and respiratory tracts, lysozyme in secretions, which is the mucus in the eye, and gastric acid in the stomach. And then by cell-mediated immunity, by the cells of the immune system. And these are derived from what we call the hematopoietic stem cells. So the cells of the immune system, or white blood cells, which we call leukocytes. And these are a type of blood cell made in the bone marrow and found in blood and lymph tissue. They're part of the body's immune system to help fight infection and diseases. And they're distinguished from red blood cells and platelets. And the types of white blood cells are granulocytes, such as neutrophils, eosinophils, and basophils, because they contain granules, monocytes, and lymphocytes, T cells and B cells. And it's worth pointing out that checking the number of white blood cells may be used to look for conditions such as infection, inflammation, allergies, and leukemia. Right, hematopoiesis. This is the production of blood cells, red blood cells, platelets, and white blood cells. Now, in the fetus, the liver is the major site of this until shortly before birth, when stem cells travel by blood to the spleen that supports hematopoiesis until vascular invasion of cartilaginous bones begins the process of ossification and formation of the bone marrow cavity. And once formed, the bone marrow is the primary site of adult hematopoiesis, although some of these stem cells in connective tissue of the abdominal mesenteries, that sounds a nice word, the abdominal mesenteries are an organ which holds the intestines to the abdominal wall, and these can be induced to form hematopoietic colonies by cytokines released during inflammation. Right, so this diagram lists the various cells produced from bone marrow stem cell, and they derived from myeloid progenitor cell and lymphoid progenitor cell. The myeloid progenitor cell leads to red blood cells, platelets, and then the white blood cells, leukocytes, which include the phagocytes, basophils, eosinophils, neutrophils, mast cells, monocytes, macrophages, and dendritic cells, and then the lymphocytes, B cells, and their associated plasma cells, which secrete the antibodies, T cells, and natural killer cells. And the B cells and T cells, because the plasma cells form part of the adaptive immune response. These cells here correspond to the innate immune response, along with the natural killer cells as well. Right, I'm going to show this video now of white blood cells attacking an invader, a small worm. Now, I hope this plays in the recording. If it doesn't, then if you click on this link here, you should be able to see it. Right, it's based on time-lapse photography. You can see the worm being attacked by white blood cells. Okay, this is speeded up. The 11 seconds shown here corresponds to 80 minutes of the time-lapse photography, but you can see what's going on. The invader is being engulfed and dealt with. Right, innate and adaptive immunity. Innate immunity, this is the first line of defense. It's relatively non-specific, but relatively immediate, fast-acting. It's present from birth, and although it's non-specific, it can distinguish between cells of the body or self cells from non-self cells. So it can distinguish an invader from self cells, but it can't distinguish between the different types of invader. And this distinction between self cells and non-self cells is done via what's called toll-like receptors. Then there's the adaptive or acquired immunity, the adaptive immune response. This is a specific response against specific antigens, but takes a longer time. It's in later development. The response can be cellular via T cells or cytotoxic T cells, and it can be molecular or humoral response. That means it's secreted antibodies into the what's called the humor, which means fluid. It's an old name for fluid. So the molecular or humoral response, and this involves B cell antibodies. And the division of the innate and acquired immune systems is shown quite nicely by this diagram here. So here's the parent stem cell into the common myeloid progenitor cell, which produces all this, and then the common lymphoid progenitor cell, which produces these. So these are molecules of the adaptive immune system. So these are cells of the adaptive immune system, and these are cells of the innate immune system. So if we consider the innate system first in some greater detail, and the so-called phagocyte, or the main phagocyte cells, these include the neutrophils. These are the major circulatory cell in the blood, 90%, and can also cross the capillary wall. And they tend to be short-lived cells lasting 2 to 3 days. Then we have the eosinophils. These are a much smaller number in the blood. They have some phagocytic activity, and they can degranulate and release toxic chemicals. And they're thought to have a specialized role in combating the invasion by large parasites. Then there's the monocytes. These can circulate in the blood and are short-lived, 2 to 3 days. They can migrate into tissues and enlarge to become what we call macrophages, which are much longer lived and can last months and years. Then there's another type of phagocyte. This is a very important type. These are dendritic cells, because dendritic cells link the innate response with the adaptive response. And these are one of 3 main types of antigen presenting cells, APCs, the others being macrophages and B cells that link the innate response with the adaptive response. They perform phagocytosis and preserve information from the ingested particles, which are presented on the surface. This can be antigen fragments, short chain amino acids, and can interact with cells of the adaptive system. So they can kickstart the adaptive immune response, and they thereby act as a messenger between the innate and acquired immune system. And the dendritic cells are particularly important because they encounter pathogens at the point of invasion. Skin, epithelia, and the epithelia, cilia, but epithelia of the mucosal, the mucosal interface of the alimentary system, and then the cilia and epithelia of the respiratory system. And then they can link with the adaptive immune cells in the lymph. So dendritic cells are a very important part of the link between the innate and adaptive response. As you can see from this diagram here, they look very different from the other cells of the innate immune system. And then we have the auxiliary cells of the innate system. This includes the mast cells. These are associated with blood vessels and tissues. and they secrete soluble mediators of immunity, such as histamine, and these were first discovered by Paul Ehrlich in 1877. So a bit of historical information there. Then the basophils, or basophils, these are relatively minor component, and they're similar to mast cells in their action, but are circulatory. And then we have the cells of the adaptive immune system. These are the lymphocytes. There's the T cells. These differentiate in the thymus. They're called lymphocytes because we find them in the lymph system and lymph nodes. So T cells differentiate in the thymus. They belong to the cell-mediated response. And there are two major types: the helper T cells, or TH, and the cytotoxic T cells, TC. The helper T cells bind to antigen-presenting cells, dendritic cells, and also B cells. They can also be antigen-presenting cells, as we'll see. And then they secrete cytokines to activate the immune system. The cytotoxic T cells, TC cells, bind to infected cells and kill them. And the B cells, these differentiate in the bone marrow, and they can differentiate into plasma B cells after interacting with the helper T cells to produce antibodies, part of these form the humoral immunity system. T cells and B cells. There's a third type of lymphocyte. These are called natural killer cells, but these form part of the innate system response. And these target intracellular viruses, and other microbes, and also cancer cells. So they're not as specific as T cells, although they can distinguish cells from invaders. Or infected cells that go wrong, as cancer cells. So why do we need these different lymphocytes? Well, this is because of the, if like, plethora of the invasions that are presented to the adaptive and innate system. And it allows the recognition of both intracellular and extracellular microbes and pathogens. But note, humoral and cell-mediated elements occur in both innate and acquired immune systems. Let's consider now the immune response. This is summarized very nicely in this diagram here. The invading pathogen, antigen, is recognized as foreign by innate immune cells, the dendritic cells and other phagocytes via receptors. Subsequent phagocytosis occurs to destroy the pathogen, and this is often a sufficient immune response to deal with the invader. If not, phagocytes can present antigen fragments on the cell surface that are recognized by T cells, or sometimes directly by B cells. And this stimulates, it sends a signal to stimulate the proliferation of appropriate T and B cells by a process called clonal selection, specific for the recognized antigens. So amplification of T and B cells specific for the recognized antigen or antigens. Helper T cells, TH cells, enhance B cell interactions and clonal selection. And then they stimulate the production of plasma B cells and antibodies from these B cells, which are directed against the invading pathogen. So the plasma B cells, the B cells proliferate into plasma B cells, stimulated by the helper T cells, and they produce antibodies against the invading pathogen. Right, let's consider a couple of examples. This one's the innate immune response to a splinter. We've all had splinters, and we've all know how it hurts, but then we also know how the body can recover from splinters. So this is what happens. So first of all, damaged tissues attract mast cells in the tissue, which release histamines, which diffuses into the blood vessels. So histamines diffuse into the blood vessels. Secondly, histamine causes the vessels to dilate or expand and become leaky. And then complement proteins leave the vessels and attract phagocytes. Now, complement proteins aid in the destruction of pathogens by piercing the outer membranes, cell lysis, or by making them more attractive to phagocytic cells such as macrophages. And this process is known as opsonization. And we talk a bit more about opsonization in the following lecture, which introduces the structure of antibodies. Thirdly, blood plasma and phagocytes move into the affected tissue from the vessels. And next, the phagocytes engulf bacteria and dead cells and destroys them. And when they're destroyed, histamine and complement signaling cease, no longer needed, and phagocytes are no longer attracted. And then growth factors from white blood cells and platelets stimulate cell division in skin cells, healing the wound. Right, so there's a related example to this. Again, it's part of the innate system. This is a video of the immune response to a stab wound. Right, let's see if this plays. If it doesn't play, then you can go to the lecture notes, and if you click on those yourself, like I'm doing this now, you can follow this video, but I hope it works directly. And if you can't hear the commentator talking, again, play this back from the lecture notes directly. So here we go. Right, let's move on to the adaptive immune response now. And let's introduce the first molecule, which is T cell, or the cytotoxic T cell, TC. This is also known as CD8. Now, CD stands for cluster of differentiation, and the cells of the immune system have these markers on their cell surface to help the system delineate one type of molecule, or one type of cell from the other. So with the CD8 or CD8 plus marker, this signifies the cell is a cytotoxic T cell. And what happens is that the T cell receptor on a CD8 or cytotoxic T cell recognizes a pathogen peptide displayed on the surface of an infected cell. And this pathogen is displayed on a molecule known as a major histocompatibility complex molecule of class 1. So this is needed. It needs to be displayed on the MHC in order for the T cell receptor to identify the antigen or pathogen. Then the T cell releases cytotoxic molecules that kill the infected cell and stop the pathogen from spreading. Now, the MHC molecule is an MHC class 1 receptor, which is recognized, a molecule which is recognized by the T cell receptors. So the antigen is presented on an MHC class 1 molecule to be recognized by the T cell, cytotoxic T cell receptor. That's how the infected cell is dealt with. Then there's another class of T cell, which is the T helper T cell. And this has a different cluster of differentiation marker on the surface. The T cell, helper T cell has a different cluster differentiation marker on the surface. This is the CD4 or CD4 plus marker. So this tells the immune system, I'm a helper T cell. And what happens here? The T cell receptor on a CD4 helper T cell recognizes the pathogen peptide displayed on the surface of an antigen presenting cell. And this could be a dendritic cell. And the pathogen peptide or antigen is presented again on a major histocompatibility complex molecule. So it's presented on this in order to be recognized by the helper T cell. And then what happens next is that the helper T cell sends a signal back to the B cells, which helps to activate them into producing antibodies. The helper T cell interacts with the B cells and then encourages them to develop antibodies. Signals are also sent out to help activate macrophages and cytotoxic T cells, and also classify Th1 cells and Th17 cells, help to recruit neutrophils and macrophages. B cells themselves, some invaders can make themselves to make their way through to the lymph system where the B cells are, avoiding dendritic cells and other cells of the innate system. And if this happens, they can be met directly by B cells and receptors on the surface of the B cells. And this can be done without the aid of major histocompatibility complex molecules. So the antigen from virus or whatever makes itself through is picked up by the B cell receptor, and then what happens is that this is internalized like phagocytosis and degraded to peptide fragments. The fragments then bind to an MHC class II molecule and then transported to the cell surface of the B cell. And then the helper T cell recognizes the complex of antigenic peptide with the MHC class II, very similar to what happens with, for example, a dendritic cell presenting an antigenic peptide, and this reactivates the B cell, and the B cell is reactivated to develop into a plasma cell and produce antibodies. So, in terms of B cell activation, each individual B cell or T cell receptor has a unique shape. And when this receptor binds to an epitope on an antigen, the NAT cell is activated. We call this clonal selection and undergoes rapid expansion. So this is the signal for the cells to proliferate in response to an antigen. And the antigens may have, on a virus, bacteria may have several different epitopes, and this can lead to the activation of several types of B or C T cells. And this is called a polyclonal response. An antigen has several epitopes. And this clonal selection expansion hypothesis, it's based on an idea firstly proposed by Paul Ehrlich around 1900 and developed by Frank McFarlane Burnett, and he got the Nobel Prize, I think it was 1960, for the development of this clonal selection hypothesis, which is shown here for B cell proliferation. And the theory says that all antigen-specific receptors exist prior to antigen exposure, therefore meaning there is a ready-made antibody diversity. And each antigen selects a pre-existing clone of specific lymphocytes and B cell or T cell and simulates proliferation of that clone. This is clonal selection and an expansion. And though the diagram shows this process for B cells, the same principle applies to T cells. Some additional points. B cells encounter the specific antigen and are stimulated to proliferate and create plasma B cells that produce antibodies. I think we just referred to that. Specific long life memory B cells are also created ready for future responses. And this is the basis of vaccination. CD4 plus the differentiation 4, T helper cell interaction with B cells is required for the activation proliferation. And regulatory signals are involved via molecular interactions involving other molecules such as CD40, CD40L, and CD80 and CD28 interactions. And finally, cytokine signals from various interleukins also play an important role. You can learn more from these links here, which I've given. So, in summary, we've learned about the main components of the immune system, the functions of both innate and acquired immune systems, the key stages of the immune response, including how antibody producing cells are created. And as I said earlier, a good resource from Elsevier showing how the innate and adaptive immune responses are closely linked is given on this link here, and there's a direct link on the Moodles. Just look underneath the link for this recording, and you'll see a direct link for this. It lasts about 15 minutes, but it's very good and complements very nicely what we've been describing here, which sets the scene now for the rest of the module, where we're going to focus on antibodies themselves and show how they can be developed for antibody therapies. Okay.